Can you all see?
Yes, ma'am.
Okay. So today in this practical class, we will cover extraction of lipids from the egg yolk. So I think all of you have seen egg and majority of you have eaten it also. So the egg basically consists of two parts. One is the whiter part and the other is the yellow or the egg yolk part. So the white part or the whitish part basically consists of two things. That is protein and water. The water is around 90% and the protein is around 10%. Whereas the egg yolk part or the yellowish part consists of three main components. First is the majority is of water, that is around 50%. Then there is lipids, that is around 33%. And least is the amount of protein, that is 17%. Now let's look upon the lipid portion of the egg yolk. So I think everybody know about the lipid, but let's brush up upon what lipid is. So lipids are basically chemically diverse group of organic compounds which are insoluble or very poorly soluble in water, but they are easily or readily soluble in non-polar solvents. If we take the example of some non-polar solvent, then we can consider it like ether, chloroform, or benzene. Now, at this point, I want to make a important clarification why these lipids are hydrophobic in nature. The hydrophobicity in lipids is due to the hydrocarbon chain in their structure. Now, if I particularly talk about the different type, let's talk about the different type of lipids. So there is fatty acids, then there are triglycerols, then there are phospholipids, glycoprotein, many more. But if we talk about the lipid portion of the egg yolk, then they are of three major categories. The first is triacylglycerols, which is around 65%. Now, this triacylglycerol are triesters of fatty acid and glycerol. Then there is cholesterol. This is a type of steroid. Everybody knows about this. Then there are phospholipids. Now, what phospholipids are? They are amphibatic molecular structure which consists of four main components. First is a fatty acid, then a platform to which the fatty acids are attached. There is a phosphate group, and there is an alcohol to which this phosphate an alcohol which is attached to this phosphate. Now if we talk about phospholipids, then this if the alcohol group changes, the type of phospholipids also changes. For example, if ethanolamine alcohol group is attached to phospholipid, then it is known as phosphodiethylamine, and it consists of 16% of the phospholipid of the egg yolk. The majority of the phospholipids are choline type. So choline is the alcohol group which is attached to this type of phospholipids, and it's 80%, and these are also known as lecithin. Then we also know that there is phospholipids can be derived from sphingosines. So they are known as sphingospholipids. One of its common example is sphingomycin, and in the egg yolk it's present in 2%. Then lastly, we know about lysophosphatidylcholine. So these are the derivative and the cleavage form of phosphatidylcholine only. Okay, then let's move forward to the principle. So here we will be showing how the extraction of this lipid part, that is around 33%, is taking place from this egg yolk. So as I have just told you, that lipids are they are not soluble in water properly, or very like less poorly soluble. So they have a preference towards some organic solvents, for example chloroform, dimethyl ether, etc. But in case of biological samples, what happens is lipids are generally associated with proteins. So they cannot be efficiently extracted by non-polar organic solvents alone. So we need some of the solvents which have some solubility in water, so that they can form a hydrogen bond and split the lipid protein complex. In the entire experiment, which we will be demonstrating, we will be using a solvent, which is a mixture of two type of compounds, that is chloroform and methanol, in a ratio of 2:1. Okay. So here,
Ma'am,
yes, please.
ma'am, lipids are found inside the cell, like only in plasma membrane, or even inside the cell, like uh, lipids found in a cytoplasm also, or just in the plasma membrane.
They are present. They are
Yes.
present.
So lipids are found in cells, like they are stored in the form of as a fatty granules. You must have heard of, and you must have also heard of adipose tissue and muscle fibers. So lipids are stored
Yes.
in cytoplasm also in these cells principally. Otherwise, they are found in all cells.
Oh, adipose tissues. Okay, and muscle cells. Okay. Okay, sir.
Yeah. Yes.
Okay, so lipids, okay, let's go to the next slide. So here we have demonstrated the basic like structure, how it is done, how the extraction is being taking place. So first of all, we will take a biological sample. So it can be blood, tissue, microorganism, or cell pellet having lipid. Here in this experiment, we have used the egg yolk. After that, we will mix the sample with the extraction solvent. In every case, it will be like chloroform and methanol in the ratio 2:1. And then we will incubate it for some time. After this, we will finally add water to it for the splitting of the lipids from the proteinous portion, and incubation will take place around for 30 minutes. After that, in the third figure, we can see that the separation has taken place. In the above part, we found salts, amino acids, metabolites. In the middle portion, we can find the precipitates of protein, and the lower part consists of the lipids. Okay. In this experiment, the materials which we will be utilizing is the egg yolk. Then we will use extraction solvent, that is chloroform, methanol in a ratio 2:1. Then we will be using sodium chloride, 0.9% concentration. Why are we using this aqueous salt is because they will help us to create a biphase system which will separate the hydrophobic lipid components from water soluble ones or the non-lipid portions. Then we will use a separatory funnel, measuring cylinder, and an oven at 40 degrees Celsius. Okay. Now let's go through the method. In the next slide, we will be demonstrating an entire video. How is it happening? But let's go through the step one by one so that after watching the video, you will have a very clear picture about it. So first of all, what we will do, we will pre-weight the egg in a beaker, and then we will crack the egg. We will separate the white portion, and we will only take the yolk part or the yellowish portion. After that, we will take chloroform and methanol in the ratio 2:1, and from that we will add 45 ml, and we will stir it continuously for 30 minutes. After stirring, we will filter it. After filtration, we will again add 20 ml of chloroform methanol mixture in a ratio 2:1, and then we will again filter it. We will repeat this step twice so that we can re-extract the lipid properly. After that, we will transfer the entire filtrate to a separatory funnel. Then we will add sodium chloride, 0.9 concentration solution at a 1/5 volume, and in order to remove the non-lipid contaminants. We will mix the entire mixture very properly, and we will put it into the separatory funnel and leave for some time so that different or two layers or phases can be formed. Very carefully, we will collect the lower part, which contain the lipid. We will repeat this step so that the proper extraction of the lipid could take place. Now we want to concentrate the amount of sample. For that, we can use like oven, we can use rotary vacuum evaporator, or we can also use stream of nitrogen. Lastly, we will weight the amount of lipid extracted, and we will term it in milligram of total lipid per gram of fresh tissue. Now let's see the video.
Actually, there is a concept going on in another process. So I will get into details like close the amount. Actually, the people are not trying to get there. People can't they're missing it. People are actually not at. So I will tell you on Thursday. Totally. Yes. Yesterday evening, I was in a car. After that, I thought to continue, but after that, I remember today. Thank you.
So we have seen the extraction of lipid through a video. Lastly, in order to concentrate the samples, we have used hot air oven. But instead of hot air oven, we can also use rotary vacuum vacuum evaporator. It is generally used for large samples. Then we can also use nitrogen steam at 40 degrees Celsius to prevent the oxidation of lipids, because in hot air oven, there might be a chance of oxidation. And finally, we found a yield of around 33.5 milligram per gram. So that's all about today's class. If you have any questions, please ask me now.
Ma'am, can you please explain again, like why we are adding NaCl?
Yeah, so we are using NaCl because it's an aqua salt solution, and this will create a biphase, means two phases will be created. So in one of the phase, the lower phase, we will find the hydrophobic lipids which we want to extract. In the other part, we will get some water soluble impurities or the water soluble lipids. Okay, so they can be separated. Only the extracted lipids can we get. So we are using this aqua salt. Okay.
Yes, ma'am. Yes.
Any other questions?
Ma'am, you can upload the slide. We will go through that and see if we have
Yes.
any other.
Okay. Give me a minute. Just wait a minute. It's uploading. Give me 2 minutes.